Assalamu alaikum. Hello everyone. I hope you are well. Let us discuss the Maranda High School mock exam biology paper 2. On to our first question. The diagram below represents a plant tissue. Okay, let's have a look at it. This is clearly the phloem tissue that is involved in translocation of manufactured food substances. So this simply means that it transports manufactured food substances in solution form from the leaves to the rest of the plant. Okay, part A. Name the structures L and O. Okay, let's look at L. There we have it. So that is a sieve plate. Plate because it's clearly pointing at 1. O. Okay, so there we have O. That is plasmodesma. Now I know we are used to referring it, or referring to them as plasmodesmata. We tend to talk a lot about these in plural, but this is clearly pointing at 1. And the singular form for plasmodesmata is plasmodesmum. Now let's briefly talk about this. So when you talk about plasmodesmata, these are tiny channels that pass through the cell walls and by doing so directly connect the companion cells with the sieve tube elements. Now, in a phloem tissue, we are going to have two main components. We have the companion cells, okay? Do you see where M is pointing at? Okay, that is the cytoplasm of the companion cell. Now, we have the companion cells and we also have the sieve tube elements, where you have the cytoplasmic filaments passing across. Now, when you talk about plasmodesmata, these are, tiny channels of communication between the companion cells and the sieve tube elements. So they allow for the quick passage of small molecules such as sugars, glucose, amino acids, and even hormones. Now, I need you to remember sieve tube elements lack nuclei, okay? They lack nuclei and actually a lot of other organelles. So they rely on the companion cells to carry out all the necessary metabolic activities of a cell. So the numerous plasmodesmata come in handy. They connect the cytoplasm of the two cells, allowing the companion cells to manage the life functions, okay, the cellular activities for the sieve tube elements. Okay, I hope that was well understood. Part B, state the function of the structure N and the cell labeled M. So let's look at N, okay? Those are cytoplasmic filaments. Now, when you talk about the function of cytoplasmic filaments, they allow for the continuous movement of manufactured food substances. So manufactured food substances literally travel along the cytoplasmic filaments. Okay, M as stated is the companion cell. So the companion cell, it controls all the activities of the sieve tube elements. And it also has numerous mitochondria, to provide enough energy for translocation of food substances. Okay, moving on to part C, give two structural differences between phloem tissue and xylem tissue. Now, when we talk about these two tissues, these make up the vascular bundles, right? So vascular bundles is a tissue in plants that is responsible for transport of substances. Phloem tissue translocates food substances, xylem, water, and mineral salts. Now, moving on to the differences, structural differences, okay? We are not talking about functional differences, but the differences that are present in the structures of these two tissues. Okay, starting with the phloem tissue, this is composed of living cells, so it's actually a living tissue. Xylem, on the other hand, it's referred to as a tissue, but in reality, it's simply made up of dead cells. And as you may have learned, these are very important because they actually, enable the xylem to perform its secondary function, which is providing mechanical support to plants. Now, another difference between these two is that xylem tissue has walls that consist of lignin, So the walls are said to be lignified. Now, lignin is a complex carbohydrate. It's a polysaccharide, and therefore it's very rigid. So it allows the xylem tissue to provide mechanical support for plants. And that is the reason when we talk about the functions of the xylem tissue, we usually set them as two. So number one is transportation of water and mineral salts. And number two is that it also provides mechanical support to plants. Phloem, on the other hand, lacks lignin. So its support is provided by fibers. Proceeding to question number 2. The following figures represent the forelimb of a certain animal species. Study them and answer the questions that follow. Okay, let's look at the 3 diagrams. We have figure 1 that represents a human hand, figure 2 that represents a bat wing, figure 3 that represents a butterfly wing. Now, before we get into the actual question, let me just briefly talk about the relationships that are present within the different structures. So if I were to start with the first one, figure 1 and figure 2. So figure 1 is of a human hand. Figure 2 is that of a bat. A human and a bat belong to the same class, and that is Mammalia. So they share a common embryonic origin. But okay, if you look at the structures, actually, if we look at the structures of these two, we can clearly see the resemblance, okay? They both have the pentadactyl limb arrangement, whereby you have one long bone. In this case, we have the humerus. It articulates with 2 smaller parallel bones, okay? These then articulate with smaller bones and so on until they terminate into 5 digits, where we end up having 5 fingers in the case of humans. So these 2 structures are referred to as homologous structures. They come from organisms that share a common embryonic origin, but because they are being used for different functions, they have been adapted for different, they have been modified to perform different functions. So in the case of the human hand, we use it for manipulation, okay? In the case of the bat's wing, obvious, it's used for flight. Now, let's incorporate the third figure, and that is the butterfly wing. So the bat wing and the butterfly wing are referred to as analogous structures. Now, analogous structures are found in organisms that are of different species, okay? Not only that, but they don't share a common embryonic origin. A bat, as stated, is a mammal, okay? This belongs to phylum Chordata, where you have the vertebrates. A butterfly is an insect, so we're talking about phylum Arthropoda. These are the invertebrates, totally different, okay? So they don't share a common embryonic origin, but they both have wings because they have been modified to perform the same function, and that is flight. Okay, that is enough of a summary. Let us get into the questions. Part A. Name the bones labeled E and G. Let's look at E. Okay, that is the humerus. G, let's look at, no. Okay, G, where is G? Found G. That is the wrist bone, right? And therefore it's a couple. Couple because it's pointing at 1. A lot of students tend to miss out on these marks because they may refer to, the answer in singular form when it's supposed to be in plural or vice versa. So this is in singular form and therefore couple. State the type of skeleton represented by figure 1. In humans, we have an endoskeleton. Part C. Name the type of joint at point Q. Okay, that is the hinge joint. This is present in our elbow, so we are able to move it along one plane only. Part D. Which 2 figures represent analogous structures? Remember, we just discussed this, right? They don't share a common origin, but because they are being utilized for the same function, they therefore, have modifications that make them appear the same, but they're clearly not. Okay, let me just briefly explain this some more. Now, when it comes to a bird, okay, It's going to be made up of bone. Remember, birds also have endoskeletons. It's going to be made up of bone covered by skin. When it comes to the butterfly, it's actually made up of chitin. Do you remember chitin? Chitin is a complex carbohydrate. It's a polysaccharide, okay? So the skeleton of insects, it's made up of chitin, and it's supported by these toughened veins. So the structures are completely different, but they perform the same function, which is flight. So superficially, they may resemble one another. Okay, moving on to part E. Give the adaptational difference between structures in figure 1 and figure 3. Okay, so we're talking about the adaptational differences that are present in, what is that? Oh, sorry. It's actually figure 2. Figure 2 and not figure 3. Okay, so we're talking about the adaptational differences that are present between a human hand and a bat wing. Remember, both of these belong to the same class, Mammalia. Now, when it comes to the human hand, we have 5 digits separated into 4 fingers, and we have a thumb, which is opposable, okay? Opposable simply means that you can touch the thumb to, what is that? Pinky finger? Yeah. Okay. Now, reason for us to have this is so that we can use them for grasping or manipulation of tools. A bat wing, on the other hand, also has 5 digits. Remember, they both have the pentadactyl limb system, but when it comes to the digits that are present, or the fingers that are present in a bat wing, they are long and spread out so as to provide a large surface area for the membranous wing, allowing for flight. Okay. Okay, let us proceed to our next question. Color blindness is a disorder caused by gene mutation and is controlled by a recessive gene. A man with normal color vision marries a carrier woman. Part A. What is the chance that their son will be colorblind? Show your working. Okay, so the very first step is, of course, identification of the genotypes of the parents. Now let's start with the male. We have been told that the man has normal color vision. When we talk about color blindness is caused by an allele that is present in the sex chromosome. Specifically, it's present in the X chromosomes. Males only have one X chromosome. So that means that this allele is going to be present on the X chromosome, but absent on the Y chromosome. So when it comes to such disorders that are, controlled by alleles present on the X chromosome, males will either have the disorder or not. Okay, there's no in between for them. There are no carrier males. Now, in this case, we're being told that the male has normal color vision. So that means that the allele that is present in the X chromosome of the male's genotype is going to be normal. We're not going to have anything on the Y chromosome because this particular allele is absent. Okay, now moving on to the female, we are being told that the woman is a carrier. Now, women have 2 X chromosomes. So in this case, what will happen is that the woman is going to be heterozygous. So she is going to have one normal allele, and the other allele is going to be responsible for color blindness. So her genotype is going to be as such. So we're going to separate these into the gametes and do our crossing, and that is what the genotypes of the F1 generation will be. So if we look at the genotypes, what are we going to observe? We can clearly see that, yes, there's going to be a possibility of having 2 sons, right? That is always the case. One son is going to be normal, the other is going to be colorblind. So what is the chance that their son will be colorblind? So we are focused on the sons. Since one will either be normal and the other will be colorblind, that means the chance is 50% or a half. Part B. Name another trait in humans inherited in the same way as color blindness. Easy. Hemophilia. Part C. Briefly describe inversion in gene mutation. So, inversion occurs when a portion of the DNA strand breaks at 2 points, rotates 180 degrees Celsius, and then rejoins, but of course it's going to be in an inverted manner. Hence the name. Moving on to question number 4. Nitrogen in the atmosphere cannot be directly utilized by plants. State 2 ways by which this nitrogen is made available for plant use. So essentially, they're just asking for ways in which nitrogen fixation occurs. Now, this is done through 2 ways. Number 1, biologically, of course, through microorganisms such as the nitrogen-fixing bacteria that are going to be present in the root nodules of legumes. Number 2, of course, is fixation by lightning. Part B, state the importance of saprophytic bacteria in the environment. So saprophytic bacteria are those that, break down dead organic matter. And by doing so, they actually release nutrients in the soil. So they enrich the soil over time. Part C, briefly explain how excessive use of agrochemicals affects large water bodies. So agrochemicals are chemicals that are used in agriculture. Very good example are the fertilizers. Now, if these are released into large water bodies, like for example rivers, they are going to cause rapid growth of water plants, okay? So these water plants are going to grow rapidly, cover the surface of the water body. And by doing so, they are going to reduce penetration of light, okay? Remember, less light is going to be able to penetrate into the lower levels of the of the river. By doing so, it's going to decrease primary productivity over time. Part D, explain how competition regulates the animal population in a habitat. Now, competition is very important. It controls the population of animals in a particular habitat over time. Now, this is based on evolution. When we talk about evolution, actually natural selection, we are talking about variations. Variations are simply differences that are present in organisms that belong to the same species. So, for example, in humans, when we talk about differences in height, in skin color, such things, these are variations. Now, variations can be advantageous or disadvantageous. Advantageous variations are those that enable an organism, to adapt better to the surroundings. So such organisms with advantageous variations will survive to reproductive maturity and, of course, pass on those characteristics to their offspring. Those that have disadvantageous variations will either die out or they will migrate to other habitats where they can adapt better. Question number 5. The figure below shows the parts of the human digestive system. Study it and answer the questions that follow. Okay. So let's take a moment to observe the diagram. Pause the video and take as much time as you need. Part A. Name the organs labeled A, B, and D. So A, that is clearly the gallbladder. B, that's our liver. Okay. D is the small intestine. Specifically, it's going to be the ileum. Now, when we discuss the small intestine in our syllabus, we talk about it being divided into 2 main parts, right? We have the first part, the duodenum, and the second part, the ileum. Now, the duodenum is the shorter part, okay? It's shorter and a bit wider. That is, the portion of the small intestine that is C-shaped. It's directly after the stomach. The ileum is the longer part of the small intestine. Now, it's also narrower and it's highly coiled. By the way, C represents the pancreas. Okay, part B, state the role of part labeled C. Okay, so the pancreas has 2 main roles, 2 different roles actually. So role number one is endocrine in nature. It involves secretion of hormones, specifically insulin and glucagon. These are two hormones that are involved in regulation of the blood sugar levels. Now, number two is that it's involved in secretion of pancreatic juice, which contains digestive enzymes. Now, pancreatic juice is released into the duodenum where it acts on food substances. Part C. Name the two salivary glands in human beings. Okay, we actually have three. So we have sublingual, submandibular, and of course the parotid gland. Any of the two will be correct. Part D. Give two adaptations of the part labeled D to its functions. Okay, so we are talking about the adaptations of the ileum. What is the function of the ileum? The function of the ileum is number one. This is where digestion is completed, right? And number two is absorption of the end products of digestion. So let's talk about the adaptations. Number one, as you can clearly see, it's long to increase the surface area for absorption of digested food substances. Number two is that it's highly coiled. This, of course, will slow down the movement of digested food, allowing for more time for absorption. Number three is that it has villi and microvilli to increase the surface area for absorption of digested food substances. Now, with every single point, notice how I am linking the structure to the function. And that is how adaptations are stated. Okay, you talk about a specific structure that this part has and how it enables that part to perform its function efficiently. Moving on to question number 6. An experiment was carried out to investigate transpiration and absorption of water in sunflower plants in their natural environment with adequate supply of water. The amount of water was determined in 2-hour intervals. The results are shown in the table below. Okay, so we have the time of day, it's given in the 24-hour format, and the amount of water in grams that is lost through transpiration. Now, when you talk about transpiration, this is the process whereby plants lose water in form of vapor to the environment. Now, these mostly occur through openings that are present on the surface of the leaves. These, of course, are referred to as stomata. So we are talking about the amount of water in grams lost by the plants through transpiration. And in the second column, we have absorption. So this is the amount of water gained by the plants through absorption. Essentially, we are just comparing the amount of water that the plants lose through transpiration and the amount of water that the plants gain through absorption from the soil. Now, if you look at the first interval, this is 11 to 13, that is 11 to 1 p.m. We have more water that is lost through transpiration than what is absorbed. And this makes sense because this is midday at this point, right? So the sun is over the head at this point and it's very bright and hot. Temperatures are high. We are also expecting that we are going to have a high light intensity. Both factors contribute to a higher rate of transpiration. If we move on to the next one, 13 to 15, okay, so this is between 11 to 3 p.m. The amount of water that is being absorbed has increased, but so has the rate of transpiration. So this continues up to 17 to 19, okay? So this is between 5 to 7 p.m. At this point, you are going to note that the amount of water that is lost through transpiration is going to be equal to the amount of water that is gained, 46 grams to 46 grams. Now, from there onwards, more water is absorbed than what is lost in transpiration. And this is obvious because at this point, we are talking about night. So temperatures are expected to be lower, low light intensities because it's night. So both of these are going to cause a decrease in the rate of transpiration. Okay, Let's proceed to part A. Using the same axis, plot graphs to show transpiration and absorption of water in grams against the time of the day. Okay, so I am going to place the graphs over here. Take as much time as you need to go through it, observe it, and so on. Once you are ready, you can press play and continue with the lesson. Part B. At what point of time of the day, okay, was the amount of water the same for transpiration and absorption? Okay, we talked about this, right? So it's going to be between 1700 to 1900 hours. Part C. Account for the shape of the graph of Roman number 1 transpiration, Roman number 2 absorption. Now, if we look at our graphs, by the way, they are broadly divided into two parts, whereby we have an increase in the rate of transpiration followed by a decrease. So as such, we are going to divide it into two time intervals. So let's start between 1100 to 1700 hours. We had an increase in the rate of transpiration. Now, this was probably because of the high light intensity and also higher temperatures experienced during the day. Now, between 1700 to 0300 hours, okay, this is from 5 p.m. in the evening to 3 a.m. in the morning, we are going to have a decrease in the rate of transpiration. Obvious, because we have low light intensities and also because, we have low temperatures at night. So both of these factors cause a decrease in the rate of transpiration. Now, when it comes to absorption of water, the curve was also similar. We had an increase during the day and then later on a decrease. So let's start. Between 1100 to 1900 hours, we had an increase in the rate of absorption of water. This, of course, was to replace the water that was lost through transpiration. Now, between 1900 to 0300 hours, we had a decrease in the rate of absorption of water due to decline in transpiration rate. Remember, transpiration is important because it also ensures that there's a continuous movement of water throughout the plant. So if we have a decrease in the rate of transpiration, we will also have an overall decrease in the rate of absorption of water by the plants from the soil. Part D. What will happen to transpiration and absorption of water if the experiment was continued till 0500 hours? Okay, so if we look at the data that we have, the last one was up to 300 hours, that is 3 a.m. in the morning. So the question is asking, if we were to continue collecting data up to 5 a.m. in the morning, should we expect an increase, a decrease, or maybe no change? Based on the trend, we are actually going to expect, a decrease in both transpiration and absorption of water. Part E. Name two factors, name two environmental factors. This was actually amended that may affect transpiration and absorption at any given time. So we have wind, humidity, atmospheric pressure, light intensity, temperature. Okay. Any first two will, of course, be marked. Part F. Explain how the factors you named in E above affect transpiration. So you can choose any factor and explain how it affects the rate of transpiration. And there we have it. Now, moving on to our essay questions. So as usual, we have two essay questions. Essay question number, the first essay question, is divided into two. So the first one is based on blood sugar regulation. So that is 12 marks. Part B, adaptations of the mammalian skin to thermoregulation. Those are 8 marks. When it comes to question 8, again, we have part A. We are talking about, factors that enable the leaf of a terrestrial plant, this is a plant that is found on land, to absorb maximum light for photosynthesis. So essentially adaptations of a plant, okay, terrestrial plant, to absorb as much light as possible for photosynthesis. And part B, describe how support is achieved in herbaceous plants and shrubs. 8 marks. Okay, so what I will do is that I am going to provide the marking schemes for you, and you can go through them slowly, take your time, understand the concepts, of course, and just go through them, look at the marking schemes, look at the key points that you need to explain if such a question was to be asked. And that brings us to the end of our lesson. I hope to see you next week.